This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 59 for November 20th, 2009. Hey everybody, it's Vincent Racaniello, and I'm here at the TWiV studio in New York City, and it's time for another podcast about viruses, the kind that may or may not make you sick. We don't talk about computer viruses on this show. And I have a great cast with me today. In my office are two newcomers to TWiV, starting with Gustavo Palacios or Palacios?
Palacios. Hi everybody.
Welcome to TWiV.
Thanks.
And Mady Hornig.
Thank you. Yes, I will. I'm glad to be here.
Both of you are professors of epidemiology at the Mailman School of Public Health here, which is our neighbor. We're at the medical center, but a separate institution, right? But still part of Columbia. Right. And you're in, I guess, the group of Ian Lipkin. That be safe to say?
Yeah, yes. We're now at the Center for Infection and Immunity.
Center for Infection and Immunity. Great to have you. Also with us today, Skyping in from Florida, is Rich Condit.
Hello, great to be here again.
Good to have you back. How's the weather in Florida? Great?
Yeah, this is a great time of year. We're talking 74 degrees right now, partly cloudy, beautiful day.
It's nice. Well, we have a good day here today, but it's not 74, right? It's more like 52. And finally, from the forest in western Massachusetts, we have Alan Dove.
Yes, I was going to say from balmy and warm western Massachusetts, but it's compared to what Rich just said, it's not even close.
Not balmy.
Well, we pay for it during the summer. So.
Yes.
We have a bunch of stories today. We're going to start with Mady and Gustavo talking about what they've learned about H1N1. I got a clue from Ian a couple of weeks ago. Said you should have them on your podcast and talk about H1N1. So I always wanted to have a representative or member of that group. This what did you call it? The Center for Infection
and Immunity.
Come over here and join us. So why don't you tell us what you're doing and what you've learned about H1N1?
Well, a couple of months ago, actually in July 17, I received a call from a colleague in Argentina that they were observing an unusual number of fatal cases during the outbreak. The start of the outbreak in Argentina, the first case was detected in May 17, and so a couple of months later, they were almost one of the top listed countries in quantity of deaths related with the H1N1 infection. So obviously there were multiple scenarios that could explain that. We were talking about differences in the viruses or changes in the viruses, or obviously things related with the healthcare, or other sources, or it could be just an epidemiological problem, because normally the cases that are more studied are the more severe, so that therefore when the rate of mortality could be affected for that. But nevertheless, I discussed with colleagues at the center, and we decided that it was a good public health study to try to define what was the reason for that high case fatality rate. And so we tried to do essentially two different things. One was try to study the complete genome of cases, from the severe cases and mild cases of disease, and then do a second study using another technology that we have in the lab to study multiple infections. So I'm going to start first to what we found with the first study. Essentially, we apply high throughput sequencing technologies to try to get the complete genome of directly from the clinical sample. In this case, we did not culture the virus and then find the sequence, but directly from the clinical sample.
So this is a swab of some kind, usually,
yes,
right?
It's a nasopharyngeal swab.
Okay.
And so we got samples from 20 of the mortal cases, and samples from a big number of normal or mild cases or ambulatory cases. And then we recovered the nucleic acid, and then we apply different techniques based essentially in 454 pyro sequences. That is a technique that allows you to get a lot of sequence information in one shot, and to try to decode if there was any changes in these viruses compared, obviously, the mild with the severe. We sequenced 28 genomes, and we didn't find any difference. There were no reassortment. The viruses were very similar to the viruses that were going on in the rest of the world. There was a notorious evolution of the virus comparing with the first cases in California and Mexico, the first sequences that were available, but nothing that could explain the virulence. There were no mutations in terms of resistance to antivirals, and so essentially we keep wondering, okay, so what could explain this high case fatality rate? So we decided to apply a second tool that is a multiplex PCR, a system that is based on, it's called MassTag PCR, and was developed at the center, that essentially allows to measure or to see the presence of, to watch for the presence of up to 30 pathogens at the same time on a sample. So we have two different panels, one that looks for RNA viruses and another that looks for DNA viruses and bacteria. And we apply those two panels to these same samples, and that's where we found a surprise compared with what had been observed in the rest of the world with H1N1. What we found was a high prevalence, or a high number of co-infections with the Streptococcus pneumoniae, and Mady will explain more about all the statistical analysis that she has done with that data. But essentially, the main result is that we saw that a good percentage of the cases, the severe cases or the mortal cases, were associated with the presence not only of the H1N1 flu, but also of Streptococcus pneumonia.
So what were the ages of these?
Well, that's a really important question, because it really, we know that, the very young and the very old are going to be at higher risk for more severe disease with H1N1 and other flu. And so given that we had excluded changes in the virus itself as being, an important factor in this particular scenario in Argentina, we really started to look at the co-infection patterns and breaking it down by age groups. And we found that in the 6 to 65-year-old age group, which is an age group which we would not normally think of as being high risk, since they're not at the extremes of age, we found that there was a very high odds ratio. In other words, having strep co-colonization, if you will, or cohabitation, we could call it co-infection, but it's just a nasopharyngeal swab. But, the presence, we would need to confirm really that it was, the full infection. Right. But the presence of strep pneumo in the nasopharyngeal specimens was highly, highly associated with more severe disease, either hospitalization or fatality.
So in these, so we don't know if that bacterium was also in the lung, right?
That's right. We don't, but we do know that it applied also in, the younger age groups, but it was really particularly powerful in this age group where the question of what, is the risk, how should we treat the patient when they come through the door with confirmed H1N1, what else should we be worried about, should we start antibiotics,
So in these fatal cases, was pneumonia, bacterial pneumonia, part of the cause of fatality? Do we know?
Well, in some of the cases, they were reported bacterial pneumonia, but in the majority of them, it was just thought that it was the direct effect of the virus.
So I don't know much about strep pneumoniae. Is it commonly carried in most people's nasopharyngeal passages?
Not typically strep pneumoniae, but
Other streptococci.
other strep
Okay.
versions are.
So the interesting question becomes, why this population? Does it have to do with the distribution of the bug?
Well, the distribution, you mean throughout geographically?
Geographical, yeah.
We don't know. And we also wonder whether there may be other factors involved, that if you are colonized with strep, you don't get colonized with other bacteria that may be more innocuous, or vice versa. That there may be other ones that are, if they're present, then you don't get strep, and then the strep is able to take over. And there are some, neuraminases and so forth that may be involved in that sort of pathogenesis pathway, but we would have to really pursue that more specifically.
In some way, the study also answered the question about the prevalence of Streptococcus pneumoniae as carriage. I mean, we studied 40 patients that are considered severe, 39 in fact, and 160 that are mild. And the percentage of presence of Streptococcus pneumoniae in the mild ones was around 20%. More or less, and the percentage present in the severe cases was 55%, approximately 55%. So it's like what we can say is that at least in this population in Argentina, during infection with H1N1, there is a clear difference in prevalence for even for carriage of these bugs.
Yeah.
Now, as this pandemic continues, you may actually be able to get a lot more data on this from other countries. I just recently was working on a story about infectious disease testing, and this came up. It turns out one of the new H1N1 tests that's available is from this company called Diatherix. They're a laboratory. And if you send your respiratory infection sample to them, they can do a panel that will subtype the influenza virus, and it will also give you readouts on everything from Pseudomonas to Strep pneumoniae. So you may actually be able to collect data on this as the diagnosis progresses.
I mean, this association between H1N1 flu and Streptococcus pneumonia is novel in the context of the 2009 outbreak. It has been known that during the Spanish flu in 1918, one of the bugs that was more associated with the more severe cases was Streptococcus pneumoniae, also Haemophilus influenza, and other bacteria in lower proportion. But I mean, the association with what is called a superinfection or with one of the normal bacteria that colonize the upper respiratory tract that could cause disease during the H1N1 infection, it's known. What is completely novel is that, and it could be due to that the majority of the studies during this outbreak had been done in developed countries, in Europe or in the US, is that this association with bacterial disease was not reported previously. I believe that this is one of the first studies. Well, there is a report from the CDC also in pediatric cases in the US, where these are autopsy cases, and essentially they found also the presence in the lung of child with severe disease, obviously, or fatal disease. They found the presence of Streptococcus pneumonia in the lung. But in all previous studies in Germany, or even in the US or in Mexico in the early part of the outbreak, there was no association. That could be due to a very quick response or give a quick treatment for antibiotics. It could be due to the fact that in Argentina the full-blown outbreak was during the winter season, when there was more prevalence of respiratory disease. Instead, in the northern hemisphere, the time of the outbreak was essentially more during the spring, so the last part.
Yeah, there was also a difference in the identification by the WHO that this was a pandemic, and then that changes who comes in and what the public health surveillance methods are for taking specimens in the first place. So that can also alter what some of what we see, because people are more worried and they report to their doctors, and the doctors will then pass the specimens along.
So are you extending this beyond Argentina now at all? Are you looking at other samples?
There's a study with the New York City Department of Health looking at co-infection patterns with H1N1.
It's obviously important to know, as you said before, if someone does come to the emergency room with severe influenza, do you start antibiotics? Is that a typical treatment to do that or not? I don't know.
It's always a concern because people don't want to get into issues regarding antibiotic resistance, and so one wants to confirm that there really is a need to to do that. And we don't know what would happen if antibiotics were instituted at the point that they come in, requiring hospitalization anyway. But we those are things that we can pursue and that would make sense to do in a a randomized clinical trial to look at that.
Last week we had Josh Stillman, who's an ER physician here, and he said they wait for the diagnostic test before they do anything. If someone comes in with influenza-like illness, they first do the rapid test, and then if it's negative and they look like they're sick, then they do PCR and they actually wait until they institute any antibiotics.
Well, something like this could change that.
It could. That's why. Yeah. Has this been published yet?
It's submitted for publication, and we are waiting.
Eagerly waiting.
Yeah.
So you have an association between strep in nasopharyngeal swabs and the severity of disease. Is there any, if I understand it correctly, is there any opportunity to follow these people up and find out whether the strep is actually integrally involved with the pathogenesis in their particular case?
No. What we have in this case was what our colleagues in Argentina were able to essentially once that they found the records of the mortal cases, they were testing more than 20,000 samples at the peak of the outbreak. This is the national reference lab in Argentina. So and what they did was, once that they identify the samples from patients that later, after collection of the samples, I mean, they became more sick, and then they have the disease, and they were fatal cases. So the only sample that was available is this one, and there is no autopsy or any other sample to follow up.
Yeah, but clearly now, when people do come in with serious flu, they should be checked, and you can do a more specific assay now. You don't have to run the whole pathogen panel that you usually do, right? So, there was this outbreak in the Ukraine recently, which we were going to talk about anyway, where there was apparently lots of fatal cases, and that would be a good place to do this as well.
I mean, when I was looking into the PROMET report for the Ukrainian, I mean, it's essentially the same kind of story that in Argentina. I mean, the first reports were high prevalence. There's nothing different, or we didn't find anything different after the sequencing with the virus, and why there is high case fatality rate. Obviously, one of the first things that we thought was in the developing world, probably the quality of the healthcare is not as obviously is not as good as in the developed countries. So it's possible that the high case fatality rate is also affected by underreporting of the mild cases. So you need to take into consideration that for the analysis.
Now, you did this study in Argentina because you have a connection, right? That's where you're from. But could you also reach out to the Ukrainian health officials and tell them you'd like to do the same thing? Would they be amenable to that?
I think those sorts of studies are great to do, but you often have to have the infrastructure already set, in order to do that. So in Argentina, the National Institute of Public Health was, collecting, and there was sort of a mandate to collect specimens, and so those were in a repository. So all of that was already set up. If there were similar systems to tap into, I think that would be fantastic.
Right.
But, it's hard to do otherwise.
Okay. Yeah. From what I've read about the Ukrainian situation, it probably doesn't exist that you can do that. Yeah. All right. Well, that sounds great. I'm looking forward to reading it when it comes out. Any other questions from Alan or Rich about that?
No.
No, it's very interesting.
It is definitely.
Thanks.
I want to let's talk a little bit about the Ukrainian situation. Maybe you guys have some insight. I got an email. I have several emails about this, and people asking me to talk about it, us to talk about it. And this one is from Arsen, who's from the Ukraine. He said, I was following your podcast for months now, so I'm quite informed about how U.S. is preparing for flu. I thought you might be interested in knowing how countries of the third world are handling this year's flu. And then he writes, a few days ago we had an outbreak in the Ukraine. So this is an old email. It's how far behind we are. This topic has been immediately politicized. In the last few years, we had a permanent government crisis. A friend of mine wrote on his Facebook page, I think that's just a new trick to say that she, S-H-E, works and she is a real medicine. H1N1 is just a new trend in Ukrainian political technologies. And this topic will be used widely this month before the elections. And he writes, she, by the way, is our Prime Minister, Timoshenko. So that's what they call the prime minister, she. The core and the fuel of the government crisis. And she has instituted price controls on medications, which has caused flu drugs to promptly disappear from drugstores. By the way, in the Ukraine, we don't use Tamiflu. Yesterday, I visited a few local drugstores, and none of the merchants knew of it. Instead, we use a local medicine called Amizon, A-M-I-Z-O-N. Today I have read a report stating that Amazon is ineffective against H1N1. I don't know what it is. I searched for it and Google said, do you mean Amazon? Of course, we also don't have any H1N1 vaccines, of which I know. But what really makes me mad is the panic. In Ukraine, everyone is in a panic. This is the first time I saw a person, actually many people, walking around wearing masks in my hometown. I have a feeling that we've got a mass hysteria where every single flu-related death is broadcasted all over the media. Also, government quarantine measures have spoiled every single Halloween party in the Ukraine. That's probably a good idea, right? I personally think that this is clearly overreaction. If we don't have vaccines, the easiest way out for everyone would be to just get influenza, get sick, heal, and be done. Postponing it would be no good. Some time ago, I watched a movie according to which if you used antiviral drugs to contain virus outbreaks, it would be ineffective. When you do that, after the country runs out of antivirals, the epidemic would proceed as if nothing happened. To me, this is similar to quarantine. Everyone sits it out, then the quarantine is eventually lifted and everything can start anew as if nothing really changed. I'm curious about your professional opinion about the measures taken by my government. Also, I'm curious about what measures you would recommend if your country was ill-prepared as mine is. It would be interesting if you did a podcast on epidemic containment. Thanks, Arson. Well, I don't know much about containment, but we could do a podcast on one because Steve Morse here would be a good guy to get, and there's some other people we could. So do either of you know anything about this situation?
Well, I don't know anything all that specifically about the situation, but rather I'd like to comment on his concern that, there's panic and people are wearing masks and that, there's something bad, about that. And in fact, during SARS, Malik Paris, who's, a well-known virologist, was noted that in Hong Kong, during the SARS era, when everybody was wearing masks and was using different types of hygiene, measures, there was a lower rate of seasonal flu. And the lowest, in fact, that they had seen in their memories. And so, there is quite a lot that can be done through, hygiene methods. And I think we should actually make it like Halloween every day. Just dress up with the masks. If somebody's, sick, it is a way to at least reduce the spray and spread.
Yeah. So, Gustavo, do you know anything about this Ukrainian situation?
No, but I don't know anything in particular about them, but it looks very similar to what I hear happening in Argentina, especially the political analysis also. I mean, everything is politicized, is political in those countries, and therefore everyone is blaming the government and at the same time not trusting the government. So and there is no institutions that take a role like WHO or CDC that are seen as sincere and honest and trying to do the good things. So that's a difficult situation because no one believes in the authorities.
Well, there were quite a few articles on ProMed Mail, and there's one here. Doctors blame the news media and politicians for spreading fear and misinformation. The mayors of cities said that the Federal Epidemiological Service refused to act without laboratory confirmation that the virus was present, delaying serious measures by nearly 2 weeks. So this was quite an extensive outbreak. I have the numbers here. 282 deaths as of Saturday in 23 regions. And what were the numbers? Over 1 million infections, I believe. 1,347,538 people have been infected. I don't know where they get the number from. I'm sure they're not
or
all...
if it's confirmed.
Yeah, they're not all laboratory confirmed, that's for sure. And these deaths are from acute respiratory distress, pneumonia. And there are very interesting anecdotes in these articles. There's an interview with a doctor who seems to me to be so misinformed. I mean, he says you should wear eyeglasses to prevent infection. Maybe it helps you avoid the people who are infected because you can see them better. He said you should never use antibiotics. He said that's the problem. But also, apparently, the healthcare system is in disarray here. The doctors don't get paid very much. There's no organization. People take home remedies, lots of citrus fruits and all this, and there's no organized pandemic response. So to answer Arson's question, what do? You have to have a pandemic preparedness. And we have that here, and you have to tell hospitals, if you have so many patients, and you can't accommodate them, what to do, where to set up areas to treat them, how to treat them. It's a very specific procedures. And in time when there's no infection, we think it's silly. But then as soon as the infection comes, it's worthwhile.
Yeah. Well, global distribution of vaccines is also a really key issue because, the countries that are producing H1N1 vaccines could also be thinking about the areas where there can be such a high case fatality and a much larger impact, and we could be doing a different sort of active or proactive distribution.
Right. So is it true that they don't have vaccine in the Ukraine? Does anyone know?
It could very well be true. And Eastern Europe in general also has a problem that dates back several decades. Their medical and biological research and entire infrastructure was very severely handicapped during the Soviet era because of Lysenko.
Right.
And there was a very, very powerful anti-scientific force at work that pretty much prevented a whole generation or a couple of generations of physicians from learning basic biology. So that's I'm not saying that that's what's causing the whole cause of the current problem, but that is certainly not helping matters, and I think that's probably where you get people like the doctor you see quoted who doesn't seem to understand how the disease is transmitted.
Right. And also production of vaccines through tissue culture methods as opposed to in eggs. We really do need to move very rapidly. I mean, it's a problem for seasonal flu as well. We have too long a wait and delay to go from the choice of the vaccine strains to the actual delivery of vaccine, and it is unacceptable.
In the 21st century, vaccine production should not rely on the schedules of chicken farmers.
It's true, but in a seasonal situation, we don't worry because they make them in 9 months and they get them out. But when the pandemic comes,
but they actually don't have such a great history in choosing the right vaccine strains because you have to choose them in the spring before. Yeah. So we could do a lot, and people are allergic to eggs, and So
if you could shorten that production time, you wouldn't have to choose until much later.
That's right.
Well, I think it's being worked on, which is good. And one of the things I like, which we've talked about here, is making vaccine in plants. I don't know if either of you have heard about this, but there are 2 companies globally that if you express just the hemagglutinin in plants, you can make particles, virus-like particles, and it's very cheap and immunogenic, and they can do that within a month. So they're being clinically trialed right now. I think that would be great. Well, sorry, go ahead, Rich.
I'll take the I'll take the chocolate.
Is it documented that this Ukraine outbreak has a statistically higher than normal mortality rate?
Personally, I don't see any documentation. I think the numbers that we're getting are unreliable. But let's just look. If we have 250 deaths out of 1.1 million, what's that rate? Is that...
That's less than normal, actually.
It's less than 0.04%, which is the global rate,
yes.
I think is calculated. So it's less than normal, less than what's observed. So there's nothing high. I think some of them were very spectacular and had very serious symptoms. If you read the ProMed, lungs swelling and bleeding. And this guy is a pathologist who they're interviewing here, and he makes a point of describing in great detail how they looked. He said, it's not pneumonia, but cardiopulmonary insufficiency and cardiogenic shock. The virus enters directly into the lungs. There is bleeding. Antibiotics should not be used. Now, he claimed that there were two viruses involved, para-influenza and influenza. I don't know what his data were for that. But what's important here is that finally now WHO has said, 17th November, preliminary tests reveal no significant change in the pandemic virus based on investigation of samples from patients in the Ukraine. So basically they've sequenced them, I suppose, and said there's nothing different there.
Yeah, but the bleeding into the lungs actually reminds me of some of the recent work that has speculated that pandemic flu from 1918 was a result, and the predilection for young adults, often male, young healthy males, was because the advice was to take aspirin, and there could, actually, we don't know what some of the other factors are that could be at play there. I think that's why we need to do.
Certainly, one of the things that's brought up in this correspondence from the Ukraine is that home remedies are a big deal. So that could be a factor.
Yeah, that's a big problem because without a good, well-developed medical system, you're going to have people going for home remedies, right? So, but the handfuls of aspirin, that could contribute.
So, addressing specifically some of Arsen's questions, it seems to me that he's talking about two extremes here. One is panic, and the other extreme is that everybody go to flu parties and get flu. And I would recommend a course that is in between those two. I think a reasoned and rational response from the government that is, pandemic preparedness and information about how to deal with flu and contain it locally, behaviors that individuals can do, is appropriate. So respond, but not in panic. And I certainly wouldn't go out and have a bunch of flu parties. I don't think that's a real good idea either.
Yeah, it's important to remember that your odds of getting the virus are not 100%.
Right. You can get through this. Yeah,
you could get through this quite easily without catching the virus. So it's not true that in the absence of a vaccine, the best approach is to go get the virus as soon as possible. The best approach is still to try to avoid catching the virus.
I went and got my flu shot yesterday at the Student Health Center, and they were selling flu kits.
What is that?
They have the flu kits have for $10. They've got a thermometer, tissues, ibuprofen, I forget a few other things, but the most entertaining component was there was a can of chicken soup.
That was for a common cold.
Well, maybe it works for flu too. Who knows?
Interesting. Flu kit. So you're okay?
I'm okay, yeah.
What did you get, the inactivated or the flu mist?
I got the inactivated.
And you have any arm soreness, fever?
Nope,
you're
nothing.
Fine.
Absolutely nothing. I'm fine, and I'm feeling just.
Has anyone else gotten the H1N1 vaccine yet? No.
I got the flu. I got the virus. So.
You got the actual infection.
Yeah.
Laboratory confirmed.
Absolutely.
Yes, absolutely.
How was it?
It was very mild. I mean, I didn't have almost any symptoms. My kid was also sick. He had a high fever and he was home. And when I was trying to sample him, I convinced him by sample myself, and so to show him how to get the nasopharyngeal swab, and when we tested, actually we both were positive, so we needed to stay home for a week.
Well, that's good to teach him all about sampling and diagnosis. That's great. Laboratory confirmed. It's good when you're in your own lab. It's the things that you can do. Excellent. Well, that's we're waiting here to get immunized. We still don't have vaccine. I think it's
I managed to get.
They're handing it out here right now. There are people lined up down in local lobby at our health science center. So we're in good shape.
And you, Alan?
My daughter got it, and my wife got it at work. They vaccinated everybody who has patient contact at her hospital. But I still can't get it. It's still not available to the general public in my age group.
Well, you're kind of an old guy, Alan, so you'll be okay. You have a lot of flu experience, so you'll be okay.
That's right.
Okay, we have one more story here to do, but before we get to that, I have a word from our sponsors of this episode, Citrix, the makers of Go to My PC. And now we're approaching the holidays, Thanksgiving and all the holidays in December, and with that, parties, shopping, time with your family and friends. But for some of us, the holidays do not mean that business stops. So if you're traveling during the holidays or you need time off, you may need to stay connected to your work. I plan on using Go to My PC to access my home computer while visiting friends. I can access any file or program. I don't need to haul my laptop with me or copy important documents to a flash drive. Go to My PC gives me peace of mind because I can access anything on my computer from anywhere. Listeners of TWiV can try Go to My PC free for 30 days. That's a month of unlimited remote access for free. For this special offer, you must visit www.gotomypc.com/podcast. That's gotomypc.com/podcast for a free trial. And we thank Citrix for their support of This Week in Virology. And our final story for today, Rich brought to our attention quite a while ago. Human vaccinia infection after contact with a raccoon rabies vaccine bait. What's that about, Rich?
As background here, I think we ought to just very briefly do both vaccinia and rabies. I know we've talked about both these before, but rabies is a negative sense single-stranded RNA virus, an envelope virus, very simple genome. We're talking about just a half a dozen proteins with importantly, one major protein in the envelope. That's the major antigen that's important to have immunity to if you want to be immune to the virus. And rabies is, in humans, a zoonotic infection. It has an animal reservoir. In developed countries where we have good animal pet vaccination programs, the reservoir is primarily in wild animals. We're talking skunks, coyotes, raccoons in particular in the U.S., and we're talking in the U.S. on the eastern seaboard and in Texas. In developing countries, the major reservoir is dogs, because the dog population is not well controlled and certainly not vaccinated, and most of the transmission to humans comes from dogs. And transmission, as most people know, is through bites. And you get bitten by an animal, you get infected at the site of the bite by the virus, and then it'll infect neurons and travel up to your brain. And once you start to get symptoms, it is, except for a couple of cases that we've discussed here, fatal. So controlling the spread of the virus in its reservoir is one of the best ways to manage the disease, which means somehow coming to terms with controlling the spread of the virus in the wild population of raccoons or skunks or coyotes or whatever. So that's rabies. On the other side of things, we have vaccinia virus. This is a pox virus, large double-stranded DNA virus, very complex. It's the virus that was used to vaccinate people against smallpox. At one time, 30, 40 years ago, everybody was vaccinated against smallpox, but since smallpox was eradicated, we don't do that anymore. So we have a population that's naive to pox viruses and vaccinia in particular. And as we've also discussed before, you can engineer vaccinia virus to express proteins of foreign origin, and this can be used to make vaccines. So in this particular case, for example, you can splice the rabies membrane glycoprotein, that's the single protein that I mentioned in the envelope of the rabies virus, splice that into vaccinia in a fashion where it will be expressed during a vaccinia infection. And then if you infect an animal with that vaccinia virus, they're immune not only to vaccinia or smallpox, but they're also immune to rabies virus. So this technology has been used for the last almost 20 years now to try and, well, not try, they've successfully been able to successfully control the spread of rabies in wild populations. Specifically, they've used a vaccinia virus that has spliced into one of its genes, the rabies glycoprotein, and importantly, the act of splicing the glycoprotein into that gene reduces the virulence of that virus. So it's quite a safe vector. And what they do is to make a bunch of this virus and incorporate it into a bait, which I'll describe in a second, and then distribute that in the wild. In sparsely populated areas, it's done by airplane. And in urban areas where it's more populated, it's done more carefully by hand. And this has been done both in the United States and Europe. The bait itself consists of a packet that's kind of like like mustard packages that you'll get at fast food joints, right? A little sort of plastic package that holds, I think in this case, it's about 2 milliliters of a solution that has the virus in it. And that's stuffed into a bait that's a rectangular sort of, rectangular sort of thing that completely coats the bait. And for raccoons, they use some sort of fish meal, and for coyotes, they use dog food. And they drop these things out of planes. And in Europe, it's very well documented that they've been able to basically clear rabies from the wild in places where they do this. And I think there's good evidence that it's been very successful in controlling the spread of rabies in wild animals in the U.S.
Hey, Rich, is there any consequence of putting vaccinia in wild animals? Does it grow? Does it replicate extensively in them?
That's a really good question. All I can tell you, I looked at that because I know that, well, obviously one of the things we're going to discuss here is that you can get an infection with this virus, and you can, it'll even cause lesions. But they did extensive testing on animals, actually lots of different animals, feeding them this virus. Sorry, my computer went nuts there for a second. Feeding them this virus and seeing if it had any effect. They could detect no lesions or no clinical signs in any of the animals. So it does, and they could see virus in the tonsils. Okay, so there's evidence for at least limited virus replication, and there must be in order for them to mount an immune response, because they have to express the protein. But apparently, it's a low level enough infection so that there's no clinical symptoms in the animals whatsoever. No lesions, no temperature, nothing. So it's apparently safe in the animals.
Now, it's interesting that you get replication in the tonsils in these various unrelated wild animal species, and you get immunity to the antigens on the virus, and yet the smallpox vaccine had to be delivered subcutaneously.
Well, it was delivered subcutaneously. I don't know what would happen if you took the smallpox vaccine and delivered it orally. And I'm not sure I want to find out. Keep in mind also that this is effectively an attenuated strain of virus because it's got this insertion in the thymidine kinase gene. Right. It is one of the things that it's not really a side issue, but they're using vaccinia virus here. And as we've talked about before, there are other options for vectoring foreign antigens that are even less virulent. You can use avipox viruses, notably canary pox, and that's used a lot in veterinary vaccines. I actually talked to some people about this in preparing for this and asked why avipox viruses weren't used in this situation as something that might be even safer than the vaccine that they were already using. And basically the answer was that this was one of the very first of these things that was ever developed. They used the technology that was available at the time. It's effective. The testing that is required to approve another vector is basically I guess not worth it relative to the effectiveness of what they're doing now. Okay, but there is the potential to do that in the future. At any rate, nevertheless, this is a replicating virus, and there is a certain possibility that a person could get infected with this, in particular, an immune-compromised person. So there have been 2 such cases, and the one we're discussing is the one that was most recent. And this involves a woman who was a 35-year-old woman who was, this is in Pennsylvania, she was under immunosuppression because she had inflammatory bowel disease, and she was taking drugs that inhibited both TNF and T-cell and B-cell responses. So she was pretty significantly immunosuppressed. And she was out picking blueberries, and her dog found one of these baits and, of course, retrieved it for her and brought it back for her. And by the time it came to her, the dog had managed to puncture the packet on the inside. And by the way, these packets are stamped with a little bit of information that says if you find this, it tells basically in as much detail as it can on the little packet what's involved, and to call, it gives a number to call. So she took this thing out of the dog's mouth, and some of the virus dripped on her hand, and her hand had been scratched because she was picking blueberries. And
Blackberries, I think.
Blackberries? Okay,
Yeah.
Blackberries. That would make more sense.
Well, it's important because blackberries have thorns.
They have thorns, right.
Very good.
Okay.
I live in
it was
Massachusetts. I know these things
Well,
now.
we have Alan here, yeah.
Yeah. So it was about a half an hour before she was able to wash the stuff off and make the phone call. So that half an hour is significant because it gives the virus a chance to actually get in through the scratches and establish an infection. She made the phone call, and the local public health officials were great. They were all over this. And from the very beginning, they watched her very closely. And she did develop ultimately a total of 26 vaccinia virus lesions on her hands. There's a nice picture in the paper, which for someone like me they're beautiful lesions. Okay, they're really classic lesions. There were issues with, they stopped her immunosuppression to start with so that she could maybe mount a better immunity, but then they became concerned with doing that for an extended period of time, and they started it again. But importantly, down the road, they gave her 2 treatments. One is vaccinia immune globulin, which is just purified immunoglobulin from people who have been vaccinated. And they also gave her an experimental drug called ST246 that was developed in response to the biodefense measures. And she eventually cleared the infection, and everything is okay. Now, it's not clear whether the drug or the VIG helped because it's anecdotal, really. But this is the 3rd time that experimental drug has been used in a vaccinia virus infection. And in all cases, the infection cleared. And the drug in animal models works quite well.
Do we know how the drug works?
Yes. Well, we know a fair amount about it. So vaccinia pox viruses have 2 infectious forms. One is what's called mature virus that stays inside the cells. The other is called an extracellular virus, which the mature virus inside the cells is a membrane thing, and it acquires its membrane through an intracellular process. The extracellular virus is made by budding the mature virus through Golgi intracellularly, so it acquires 2 additional membranes in the process. And then that migrates to the undersurface of the plasma membrane, and the outermost of those 2 new membranes fuses with the plasma membrane, so that you've now exocytosed a particle that is the original membraned particle with 1 more membrane. There are several virus proteins involved in making that extracellular membraned virus, and this drug, ST246, targets one of those proteins. We know this because you can make viruses that are resistant to the drug, and they map to that gene. You can make knockouts of that gene, and it makes a very sick virus, but that sick virus is resistant to ST246. So interestingly, the drug affects spread, not replication of the virus. But in a number of animal models, it's very effective.
So what did they do anything with her dog?
Well, he's up to date on his rabies vaccine.
That's true. I'm just
there's nothing in here about the dog.
wondering if the vaccine replicated in the dog or whatever.
As far as I know, they didn't. Well, certainly there's nothing reported on the dog.
Yeah.
Based on what we know about how the vaccine works, I would assume that the dog came through it just fine.
Yeah.
But by the way, the other case that was reported was from 2007, and it was essentially the same scenario, and that is a dog bringing the bait to the owner. And in this particular case, the owner had a dermatitis, which is another and was also pregnant, so probably mild immunosuppressed. In that case, they only developed a couple of lesions that healed spontaneously, and everything was okay.
So the moral here is not to touch the bait, right?
Don't touch the bait, right? And read what's on the package. All the numbers.
Well, if your dog brings you something, the natural reaction is to take it, right?
Right, absolutely.
And then, this is virus later on. But
Maybe make the dog spit it out on the ground first and see what it is.
How do you do that, Alan?
I don't know.
So I think it's important to point out that there have been, it says since 1990, approximately 100 million doses of this animal vaccine have been distributed in the United States by hand or by airplane. And talking about how much of this sort of problem they've had. In 2008, among 16 states where a little over 10 million baits were placed, there were 291 vaccine-related telephone calls involving human or domestic contact. So that's a rate of 2.8 per 100,000 baits. And they have a very robust program to monitor all this. So that's an enormous campaign, and just 2 actual infections, and both of those were in people who had predisposing conditions, and both of them were resolved satisfactorily. So I think this is an enormously successful program.
Sounds safe. It's very safe too.
Yeah.
So do they do that, say, in New York City, in Central Park? Do we know?
That I can't tell you specifically, but I think New York State is one of the states on the map.
Because we have rabid raccoons in New York City.
It wouldn't surprise me at all if they.
Is that a dig against New York City? How do we know this is working for rabies control? Do they do serosurveys and?
My recollection, I don't know I'm just familiar with one article in Europe, and I can't tell you exactly. I do know they've somehow surveyed the population. I don't know if this has to do with the rate of reports of sick animals, or the rate of reports of cases, or whether they've actually gone out and done serosurveys of the animals. I don't know that exactly.
I think in some states they've actually done surveys, trap animals, and survey for rabies incidents.
I guess it's working, but I think that it's a great example of using technology to accomplish something.
Well, and one other interesting note: this woman has now neutralizing antibodies against rabies.
She does. She's protected.
But interestingly, she never made any IgM to vaccinia.
Right.
So there was funny business going on with her immune system.
Which is not unexpected, right?
Right,
yeah.
Absolutely.
Gustavo.
So is known if there is, in the wild population, if there is secondary infections with the vaccine? I mean, if there is replication of the virus, can the virus spread from the infected animal?
In laboratory situations, they looked for spread from an infected animal to other animals, and they saw none. So the suspicion is that there won't be any spread in the wild.
Well, it sounds pretty safe, but maybe it's not a bad idea to go to an AVPOX, right?
Ultimately, if it's going to be an investment issue. It's going to be a money issue ultimately.
Yeah. You have to prioritize.
As an aside, I looked up a review on the use of recombinant poxvirus vaccines. We talked about the AIDS trial recently where they used a canary pox vectored vaccine. That's the only sort of example I know of. I mean, there's been lots of testing in humans, but there's no licensed vaccines in humans. But in veterinary practice, it's pretty widely used. So in terms of poxvirus vectored vaccines, there's one for equine influenza virus in horses, one for West Nile in horses, Newcastle disease in poultry, avian influenza in poultry. There's 3 different rabies vaccines for cats, and the one we did for wildlife, feline leukemia in cats, canine distemper in dogs, and other fur animals, it says. So it's pretty widely used.
Hey, all thanks to Enzo Paoletti, right?
Enzo Paoletti.
And Dennis Panicali.
Dennis Panicali, that's right.
Great. All right. Well, this is 100 million doses is not change. That's a lot of money. So someone's making money.
It's a robust program, yeah. So, actually, who's making money is Mariel. They're the manufacturers of the vaccine.
Okay. It's a nice story. I like that. Well, that does it for our stories. And let's see if we can do a couple of emails before we wrap this one up. And I saved an email from Nick. Nick is a retired McGill professor who happens to have written a virology textbook. And he wrote a long email. I'll try to summarize parts of it. And he has some... we saved this for you, Rich, because we didn't get to this the last time we were on the show. I'm a bit behind in my TWiV listening, but I'd like to comment on something you said in TWiV 47. You have reminded us several times in the past that viruses are not alive. And you embellished on this during TWiV 47 by saying that if they are not alive, they can't be killed. Actually, viruses can be killed by treatment with formaldehyde or Clorox, boiling, or irradiation with X-rays or UV light. These are commonly applied treatments that kill viruses. Were they alive before having been killed? Perhaps that is just a semantic question, but it seems to me that you cannot kill something that is not alive. I think there may be some confusion in our minds about what a virus is. We most often think of viruses as being the virus particle or virion, which carries the viral genome, wrapped in a protein and sometimes a lipid coat from one cell to the next. This looks inert and not living, but this is a bit like saying that an oak tree is an acorn, or a sunflower is a sunflower seed. Viruses actually exist not only in the form of virus particles, but also in their intracellular forms, during which they carry out the many intricate and complex activities that lead to their reproduction. Sure enough, they need lots of things that cell provides, including energy, the machinery that makes proteins, and basic building blocks such as amino acids and nucleotides. But if you mean that being alive implies coding for those proteins and RNAs needed to synthesize proteins, produce energy, and make amino acids and nucleotides, this becomes a bit of a technical argument. And he goes on to say, what about obligate intracellular parasites like chlamydia and rickettsia, which are considered alive, but they need to reproduce in a cell? So he argues that viruses are just as alive, simply that they have less of the equipment of life and depend more strongly on the host. And he's not comfortable with the notion that viruses are not alive. They're certainly part of the biosphere of living things, and they share with other organisms the basic mechanisms of replication and expression of their genomes. And he ends up by saying, if you can kill it, it must have been alive.
Or as the governor said in the movie Predator, if it bleeds, we can kill it.
I like that. He said, I just listened to TWiV 48. Your further discussion of this topic was interesting, but sort of stalled. I got the impression that Rich agrees with me. Regards, Nick. All right, Rich, do you agree with Nick?
Yeah, I basically agree with him. I have a little bit of an issue with this that I'll go into, but I decided some time ago that my own personal definition of life was the evolution or perpetuation of nucleic acid sequences through time. This really all comes down to what your definition of life is, and that's why I think it's such an interesting discussion, because it challenges what our definitions of life are. And the classic definition, I think, says that you have to have your own metabolism, in which case viruses kind of fail that qualification, which is, I think, why a lot of people don't consider them to be alive. But I like Nick's argument that we're just talking about a couple of different forms. Yeah, when it's outside the cell, it's just a chemical, but that's kind of like being a seed. When it's inside the cell and parasitizing the organism, that's much more like life. And what's important to me is that it's got all the evolutionary characteristics to have its own family tree and everything else. So I think of them as alive. The problem is that if you then adopt a definition of life that includes viruses, then you have to think about some other things. And the first thing that comes to my mind is, well, not so much prions necessarily, but transposons. Okay. They are nucleic acid sequences that evolve independently of their hosts over time and move around and are amplified and et cetera. Are they alive? Now, I don't typically think of them as alive, so I'm contradicting myself.
Well, what about
crystals?
What about these small replicating RNAs, viroids? You don't even code for anything. By your definition, they would be living.
By my definition, they would be living. That's right.
But then on the other end of this, if you say that viruses are not alive, then you have to make an arbitrary line elsewhere. You're just doing it further upstream. And you would say, well, are mycoplasms alive? And at what point does something have to be, it is just being an obligate parasite enough to make you not alive?
Yeah.
So in a way, the most interesting thing about the whole discussion is that after all of this time, we don't even know what life is.
It's whatever you make it.
Yeah.
But I think this is a running problem in biology. It comes up when we try to define species too.
Of course. Let's see what our guests think about this issue. Do you ever think about this?
Well, I think of it as an ecological issue. I mean, if you take the seed, or the acorn, if you will, I mean, that is not alive, but it has the potential to be alive given the right environmental context. And similarly, one can think of a virus in the same sort of way, depending on where it is, what type of environment it finds itself, it is able to to have an effect on the organism, but it itself does not become alive. It becomes part of a host, and it can become part of the host genome, but it itself remains sort of more like the seed, and interacts with the environment of the host, the milieu of the host, if you will. I kind of like that, because that definition also allows you to look at the obligate intracellular pathogens as being parasites as being also dependent on their environment as to whether they can actually do something. They actually have the machinery, but they can't actually reproduce unless they have have the right environment.
What do you think, Gustavo?
I think that is an infinite question, because everybody is going to have different opinions, and I always thinking, okay, what are we trying to do to assign the viruses to be alive or non-living things? What is the goal here? And I think that a lot of times we became confused because we are trying to see if they have will or not, and clearly that is very easy. That they don't do anything by themselves. They are not trying to do anything. They have no will. They just are entities that do what they are programmed to do.
Yeah. No, I think that's part of my issue. They cannot do anything on their own, whereas a seed can grow into a tree on its own. You just add water, right? This seed is not living, but it can live on its own. A virus needs the cell. A prion needs a cell. A viroid needs a cell. And it's just a programmed series of events that lead to its reproduction completely independently of its own will, if it could have any will. I think the point of it is, you're right. It's about the discussion. It makes us think about what is life and how you define it and how things fit into it, and that's only good in the long run. And there's not going to be an answer.
So why don't we take a survey of the listenership?
Okay.
You should put a poll on the website.
Yeah, you can use, well, there's a number of things that you can use to put a poll on the website.
Survey Monkey.
Right.
Yeah, sure. We'll do that. We'll put a poll up and you tell us, so what's the question, Rich? Do you think
Are viruses alive or dead? Right. And you could, I don't know if you might leave room for comment. You did, TWiV 1 was on this, right?
Yeah, we've always touched on it. It's an interesting issue.
It keeps coming up.
And everyone's got an opinion, which is great. That's when things get interesting, when everybody thinks something different. I happen to have a very extreme view, and there are other people that agree with me. There have been articles written about it as well, and both sides. But I think we can touch on it all the time. I think
it's a great question to keep discussing. But I'd be interested in a poll and in the opportunity for people to comment again on the poll.
Okay.
Yeah, give them options. At least give options of living, non-living, or none of the above.
Well, look, if Arnold said if it bleeds we can kill it, then viruses don't bleed, so they're not living. That's the end of the story.
Maybe we ought to get Arnold on TWiV.
Yeah, well, I think he's too busy. Well, let's do one email more, because this is actually, you got Mady and Gustavo can comment on this, because this has to do with influenza. This is from Ariel and Ayelet in Israel. And thanks for a great podcast, one I am always waiting for. I recently got a Grippal S1 seasonal flu vaccine, and about 80 hours later I began to experience mild flu symptoms: headache, sneezing, muscle pain, runny nose, etc. I read in the vaccine manual that Grippal S1 contains influenza virus surface antigen inactivated with formaldehyde, so this virus can't replicate in the body. From this fact, I conclude that all of those symptoms are caused by the immune system response to the antigen in the vaccine.
Or a totally different virus.
Right.
Which could happen, right? Things happen when you get immunized.
Absolutely.
So I presume this is the inactivated vaccine. Do you know that Grippal? Let's see. There's a link. There's a link
sounds...
here. Oh, she sent us the inactivated, yes.
Yeah.
And she said formalin too, right? And this is actually the seasonal vaccine, H1N1, H3N2, and B, influenza B. Yeah, the symptoms are, you're making an immune response, right? And that's why you get those symptoms. So then they ask, do all symptoms and damage in influenza pathology, are they due to the immune response?
Well, that's an important question. I think we do. We certainly know with SARS, the cytokine storm, was a really important part of the immunopathology and the disease that one saw. So
right.
But in this case, 80 hours is a little bit long, unless you're thinking about a delayed type hypersensitivity response, which is not reporting that there was some type of swelling or pain at the injection site. And so that would make it less likely to be that sort of thing. It looks mainly to me like an intercurrent infection with something, with something else.
Okay, next question. Does a mild or severe disease related to cytokine storm? I think you just mentioned that.
Yeah.
Maybe in some severe cases. But my view is that that's a hypothesis, right? It's not entirely proven that a cytokine storm is responsible for severe disease. Right?
Well, but it's something we could study.
Yeah.
We could, at least if the cytokines are become systemic, it'd be hard to get a lung sample from somebody who's in the midst of illness. But if it was also peripheral, which would also relate to the myalgia and the sort of sickness behavior that goes along with that.
So they've looked at it in animal models, and that's
right.
You can do that, but to get people to have PBS put into their lungs would be harder, I would think.
Absolutely.
Next question. Does suppressing or reducing the immune response relieve symptoms and damage and can prevent opportunistic infections like pneumonia? I don't think you'd want to immunosuppress during influenza, right? It would make it worse.
Yeah. There were some treatment with corticosteroids.
Yeah.
And
what does that do? What's the outcome?
Well, there is conflicting reports. Some of them says that it improves. In other cases, it was associated with severity, so it's hard to say.
Yeah, and I think that also we have not been routinely screening for other infections. So if you have a bacterial co-infection, immunosuppression with corticosteroids or other measures would be a very inept thing to do, and and would be very risky.
Yeah, that actually. I don't want to go back to our story, but I mean, it was one of the questions. It's like, okay, you are detecting these 2 bugs, but who was first? It was strep pneumonia was first, and then you got the H1N1 co-infection, or it was the opposite, and it's a super infection, and that could be it. It could be that the sequence of events could determine if the severity.
Right. What is the pathogenesis of influenza in immunodeficiency patients?
Well, in these cases, there is an association. One of the risk factors that was found in the previous studies that I mentioned was with pregnancy and diabetes, and so it's like all those associations are related with the status of the immune response. If you have a normal, very active and fighting immune response or not.
Yeah, I think it's a very important concern, and I think has guided our current policy of trying to direct vaccine to women who are either considering becoming pregnant or who are pregnant.
Do we know in HIV patients what kind of course there is? Is it more severe, or we don't really know?
I don't know specifically in HIV, but I think immunocompromised in general.
In general, yeah. What are the chances that I got a cross-immune reactivity against swine flu H1N1 by this seasonal vaccine? 0, right? There's no cross-reactivity. It's a couple of papers have shown there's no cross-protection with the seasonal H1N1 and swine flu. But if you got the 1976 swine flu vaccine, then you're protected pretty much. So there's some upside. Only 40 million Americans got that, and I was not one of them. Finally, the virus DNA was inactivated with formaldehyde. Does the vaccine contain any DNA or formaldehyde or just glycoproteins? I'm sure the answer is no. There's a little bit of formaldehyde, mostly glycoproteins, no DNA. It's probably a little RNA, viral RNA. But the virions are purified as they come out of the egg on sucrose gradients, and they're disrupted with detergent and inactivated with formaldehyde. And so there's a little bit of sucrose, formaldehyde, detergent, mostly glycoproteins, and a little RNA. So those are the answers. Okay.
And there can be other preservatives in there too. But a delayed response such as this is unusual for something directly vaccine related.
All right, that brings us to our picks of the week, and then we'll wrap it up. And let's start with Alan.
Well, my pick this week is something that I'm wearing as we speak. It's maybe an odd pick for a science podcast, but I know a lot of people have the problem of trying to interface their sound system with their computer, especially if you're using Skype to make phone calls. And I discovered this headset. It's the Heil ProSet Media headset.
Is it USB, Alan?
It is USB. It is, well, I have it interfacing through an iMic USB port, but you can it comes with 1/8 inch standard phono plugs on the end. The one caveat with that is that the plugs for it, the microphone in is a microphone line in. So if you have a Macintosh, the line in port on it is a line level input.
Right.
So you actually have to go through a USB interface in order to get it in. But it's about $100. And it's an over-the-ear, very nice, very comfortable, proper-fitting headset with pretty good sound quality. It's a whole lot less expensive than the very high-end studio equipment that Vince has. But
yeah, Gustavo's wearing one right now. That's about $400 there.
Wow.
Okay.
But Alan sounds very good. And I'm thinking I need to get a second one because whenever we have two people, I have problems with mic cords bumping into the microphone, so maybe we'll get this one. Although I don't like the 1/8 inch jack; it tends to be noisy. We have XLR connectors here on this But maybe I'll get Dick to go in with half, and we'll get another one.
Yeah, and if you go in through the iMic adapter, Griffin makes this adapter. It's called an iMic.
Right.
Of course, it's a Mac attachment. It's got to have an I in front of it. And it allows you to select line level or mic level inputs and go through a USB port, which takes care of a lot of noise issues as well.
Yeah. Well, you do sound good, I got to admit, but
Well, thank you.
maybe it's your voice.
He does have quite the radio voice.
You do too, Rich. What do you have a headset, Mike?
Yes, I do.
It's not
Got a Plantronics headset.
Yeah, Plantronics is a good one. It's about $40, right?
Yeah, I have one of those too. I found it just wasn't comfortable for long sessions.
So what have you got, Rich?
I got a book for you.
All right.
It's called Longitude: The True Story of a Lone Genius Who Solved the Greatest Scientific Problem of His Time. And it's by a woman named Dava Sobel, or Dava, D-A-V-A, last name S-O-B-E-L.
Yes.
So this is about how to figure out, if you're on an ocean-going vessel, what your longitude is, where you are relative basically to the equator, east-west wise. You can figure out where you are north-south wise by just watching the sunrise, I think, or something like that. But longitude is a big problem, and it has enormous political and economic impact. Now, we're talking about days before they couldn't figure this out. If you wanted to sail from England to some island to discover treasure or grow tobacco or conduct a war, and you had to be at the right place at the right time, knowing what your longitude was of very significant strategic importance. And so, in fact, in Britain, and I forget what the date was, but in Britain, they were so concerned about this problem that they had a contest and gave a huge prize to anybody who could solve the problem. And there were lots of different solutions that were proposed, but ultimately the solution was a good clock. Right? Because you basically, if you know what time it was when you left, and you've got good tables of sunrise and sunset, and you can just know when the sunrise and sunset time, what time it is where you are, you can use your tables to figure out how far east or west that you've traveled. But at the time, making a clock that would do well on a boat that was bouncing around in salt water was a real challenge. And this amateur did it. And then there's all sorts of politics about how they didn't want to give him the prize and that kind of stuff. But it's a great read.
And it's
really
the story of that, basically, right?
Yeah, it's the story of that.
I think Dick has mentioned this once before, this whole idea about having a good clock, yeah.
Yeah. As a substitute, as a supplement to that, if you go to the Greenwich Museum, they've got a bunch of this guy's clocks.
Oh, cool.
And they've got a whole display of the evolution of this clock.
Nice. Great.
And he is also the reason that UTC, Universal Coordinated Time, is the time in Greenwich, England.
Is that right?
Yep.
Yeah, that's a good time to use because nobody knows what Eastern Standard Time is in Europe, right? UTC is the way to go. Great. That's great. Thanks, Rich. My pick is a iPhone app called Microbe World app. And this is actually produced by the American Society for Microbiology. It's $4.99, and it allows you to access a lot of their content directly from your iPhone or iPod Touch. You can get podcasts. You can get Meet the Scientist. You can get This Week in Virology, which is us, Microbe World Video, This Week in Parasitism, Mundo de los Microbios con Gary Toranzos. Is that okay, Gustavo?
Yes, that's okay.
Microbe World's weekly Spanish language podcast. Yes. You can also get Microbe, the news magazine of ASM, and a lot of other things. So if you like to easily get all this content from ASM, check that out. It's really nice. I bought it, and it's really quite good and well done. $4.99 at the iPhone App Store. And that'll do it for another episode of TWiV. If you're a new listener to us, please subscribe in iTunes. It's free. You can get automatically the latest episodes. If not, go to twiv.tv, our website. We have show notes, and you can play the episode there or download them directly. TWiV is part of microbeworld.org, the ASM site, and we're also part of sciencepodcasters.org and promednetwork.com, websites where you can find high-quality science podcasts from a wide variety of fields. You might also want to check out our latest podcast, TWIP, This Week in Parasitism, at microbeworld.org/TWIP. As always, send us your questions and comments. You can send us an email at twiv@twiv.tv or call up Skype, TWiV Podcast, and leave us a voice message. Gustavo, thank you very much.
Thank you for inviting me.
Really pleasure that you took your time. Also, Mady, thank you very much for coming over.
Oh, I really appreciate you inviting us.
It was great to hear your work, and maybe you'll come back another time and tell us what else you do.
Yeah,
that would
sure.
be great.
Great.
Alan, thank you very much.
Always a good time.
Dovedocs.com
Yes.
is where you can find Alan's musings from the western edges of the Massachusetts forest, full of rabies-laced bait. I
No doubt.
Go out and find some.
I'll have to.
You know, and send it to me. I'd be really interested to see. I wonder if you can send those through the mail.
Wear gloves.
I don't think you should send it, but you could, as you know. And Rich, thank you for joining us again.
You're quite welcome. I enjoy it all the time.
From
It's great.
Southern Florida. Thank everyone. Thanks for joining us. We'll be back next week, of course. Another TWiV is viral.